This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM.
This Week in Microbiology, episode number 14, recorded August 19th, 2011. Hi everybody, I'm Stanley Maloy, sitting in for Vincent Racaniello for this episode of TWiM, the podcast that explores unseen life on Earth. Joining us today from the Medical College of South Carolina, Michael Schmidt. Hi Michael.
Hello Stanley, how are you this afternoon?
I'm well, how are you?
I'm doing good.
Also joining us today from Small Things Considered in San Diego, California, Elio Schaechter. Hi, Elio.
How you doing? Hi.
And Margaret McFall-Ngai from the University of Wisconsin. Welcome, Margaret.
Hi, Stan, Michael, and Elio.
Before we get started, there's a couple of really interesting things in the news. There's always interesting microbiology in the news. And a lot of times it's picked up and reported on the website for Microworld. I thought a couple of those things are pretty cute little stories that might be fun to mention briefly. The first one is about a symbiosis in Mealybugs. And the concept here is that there are bacteria living inside of bacteria, which is a pretty interesting concept. It's almost like the concept of mitochondria inside of eukaryotic cells.
Stan, are they in the periplasm?
No, they're inside. They're in the cytoplasm.
Inside.
Yeah, they're not like...
Not like Vibrio.
No. And this is actually, the story goes back a ways because one of the authors of this paper is Carol Van Dolen at Utah State, and she found some years ago that this was the case. But now she's got an explanation, and the explanation is that one of the bugs feeds the other amino acids. So it's a two-way symbiosis that in itself is a symbiont inside of a mealybug. One of the bugs has a lovely name, Moranella endobia, and that's the reason that caught my eyes, because Nancy Moran is the person who studied this kind of symbiosis as much as anybody and deserves this honor.
Yes.
So it's pretty cool little story. It's a different way of thinking about bacteria and how they grow. The other story that I thought was pretty cool was titled Bacterial Kamikazes. And this is interesting because it's the field of synthetic biology where they've tried to engineer E. coli to kill Pseudomonas in biofilms. And in the process of doing that, the E. coli commit suicide. So they're kind of like bombers that release their target on whoever they want to get rid of. It's an interesting story, has a long ways to go, but I think it ties into this concept of how critical microbiology is for so many other fields, and particularly for this growing field of synthetic biology. With that, maybe we should get started. Before we begin, the first paper is focused on a fungus, and I should point out to all of you that our very own Elio Schaechter is one of the world experts in mushrooms.
No, come on. But I did write a book on the subject called In the Company of Mushrooms. And I am an amateur mycologist. I go out in the woods and look for mushrooms, and I still am living to tell the tale.
It's a very wonderful book, but in it he explores a lot of different kinds of fungi and the visual manifestations of fungi as mushrooms. So this first paper is on a kind of fungus that doesn't form mushrooms, but the paper is titled Non-Lytic Exocytosis of Cryptococcus Neoformans from Macrophages occurs in vivo and is influenced by phagosomal pH. This is a paper from a group associated with Arturo Casada-Val at the Albert Einstein College of Medicine and in collaboration with a group from the University of Brazil. Elio, would you begin to take us through this paper?
Okay, gladly. The story is quite interesting. I'll give you the punchline now. They and a group of investigators in Birmingham in 2006 published back-to-back papers in current biology on a finding which is really quite startling, namely that macrophages ingest this fungus, Cryptococcus, which is a yeast, and then spit it out. They actually go, ptoi! Out it goes. And both survive. Both the Cryptococcus survives and the macrophage survives. It's like they tried a bad meal and neither of them liked it, and they decided to part ways. And this is called, has wonderful names like Vomocytosis, as in vomit, Exocytosis, and so forth. Anyhow, it's a new phenomenon, and it hasn't been followed up all that much until this recent paper. But what do you think if I give you a little background about the Cryptococcus?
Great.
I think that would be great because, it's one of those unknown yet, pretty dangerous fungi that gets into a person who's immune compromised.
Exactly. This is a fungus that exists worldwide in soils, apparently transmitted via pigeon droppings, inhaled, and in immune-compromised people, as Michael just said, it causes a meningoencephalitis. A lot of AIDS patients die of that. So it's quite serious. It's said to kill 600,000 people a year. Now, that is mainly HIV patients, or AIDS patients, I should say, but it can affect 20% of the cases are in immune-competent people. So it's a dangerous bug, and they say it's quite deadly. In addition, there is a new version of it, a new species called Cryptococcus Gatai, Gatti, which kills healthy people, again, immune-competent people. It's been found in the Pacific Northwest, to talk about this continent, and apparently it's quite a killer. It's a rare infection, but it still matters.
And that's one of the things you shouldn't do if you go to the Pacific Northwest is to tramp around in the woods, because it literally falls out of the trees. And it's pretty frightening up in that area of the country, because the incidence rate has really gotten pretty high. And you can check that out at Morbidity Mortality Weekly Report. They actually have had some stories on that new variant of Cryptococcus. So if you're going to go out hiking in the woods in the Pacific Northwest, take a look at that so that you can protect yourself, or at least watch out for the symptoms.
Or use a mask.
Or use a mask. That's even better.
Walking through the woods with a mask, you could scare some people.
May have to do that.
One of the questions I think that this raises is those 20% of the people that are, in theory, not immunocompromised. It's possible that they are immunocompromised in some way that we just don't recognize.
Right. That's right. That's true for an awful lot of other opportunistic diseases and opportunistic organisms. There's a lot we don't know yet, isn't there?
Right.
But now, the organism is kind of unique. It's a yeast. It belongs to the Basidiomycetes. In other words, belongs to the mushrooms. It's not an ascomycete, which is yeast and most molds, and to which most pathogens belong. This is a Basidiomycete, but it doesn't make a fluting body. Let me just tell you, I got to slip this one in. If it were to make a fluting body, by analogy to what it's close to, it would be a thing called witch's butter, which is a little sort of yellow concretion that grows out of trees. So it would look if it could do it, it would look kind of funny too. Anyhow, this is a yeast that's surrounded by a big capsule, and the capsule is a virulence factor. But the capsule does not keep the organism from being phagocytized. So when we inhale this organism, the macrophage in the alveoli take it up, and in some mysterious way, may or may not play a role in the transport. By the way, a digression I want to make. I don't know if I made this before, but don't you think it's kind of weird that some of our cells are outside our body? Like the alveolar macrophages are topographically outside the body. Right. They come in and out. That's a different concept of the human body as being separate from its environment. Don't you think?
It is a really intriguing concept because there's a definite selective advantage to send these, if you will, soldiers out to police the area to make certain that anybody trying to invade us, and I'm anthropomorphizing this into, using an army to keep another army from, getting a foothold in our sterile environment. And, that's the way I've always looked at it, is you put these things out there. And, I'm a very simple microbiologist, and I always thought the human body was like a donut. And, the inside, or our insides, is actually like the hole of the donut, and you have to sort of penetrate that Krispy Kreme glaze to get into our sterile tissue.
Is our tissue really sterile?
Right.
Is it really sterile? So I asked Marty Blazer this question, and he said that the jury's still out. I wanted to know if blood was have people taken blood and looked with universal primers? Looked for the vestiges of bacteria in blood?
Well, that's an interesting question. We do know that we have showers of bacteria entering the blood when we brush our teeth too heavily, or when we get dental or teeth cleaned. But that's transitory. You're asking. So in a transitory way, the answer is yes. How permanent the bacteria are in the blood that's a good question.
That's a little off topic, but I was just curious.
But so I don't think that's off topic at all. I think it's right on topic. To me, when you start thinking about our microbiome as being a part of us instead of just something we drag along, then our whole way of imagining our body of having this inside and outside is convoluted. So then I think it makes this idea of macrophages transiting between the two parts of our body to make a little more sense.
Well, so now a macrophage is taking up a Cryptococcus. What happens is that the macrophage finds the yeast finds itself inside of a vacuole, a phagolysosome vacuole, which arises by diffusion of lysosomes, which are bags of deadly enzymes, fusing with the phagocytic vacuole. Now, in most cases, this is the end of the parasite, but Cryptococcus does fine. It just survives that dandily. And I believe, Stanley, isn't this true for Salmonella also, which is kind of unique in being able to survive in a phagolysosome?
Yes, Salmonella loves phagolysosomes. It's like the briar patch for Salmonella.
Wow. Anyhow, so the idea is that how come it does that? Well, there are antioxidants, and one of the antioxidants that Cryptococcus seems to make is melanin. Now, let me go into a real digression. Is that okay? Here's the story. Chernobyl, after the accident happened, the people, the technicians there, sent in a robot into the chamber containing the blown-up reactor, and they found that the walls of the reactor on the inside were black. Now, how come they were black? With an incredible amount of radiation, they took samples, brought them out, and they're full of fungi. Meaning a fungus which is common in the air, a mold called Cladosporium, but also Cryptococcus. Now, get this. Arturo Casadevall, the same author as this paper on exocytosis, found that some of these fungi actually used radiation as an energy source. Now, if you think that's wild, think a little bit further. This is part of the electromagnetic spectrum, and so is light. And light organisms use an antenna called chlorophyll. In the case of the fungus, the antenna for capturing radiation is melanin.
Oh, wow.
And sure enough, albino mutants of that fungus, I told you about Cladosporium, do not use radiation. Now, to me, this is such a fundamental story and such a paradigm shiftlet, at least, and it hasn't really been followed up. And
that is so cool. Oh.
But it will actually follow the concept that was just advanced of the electromagnetic spectrum being harvested by microbes. We're going to take up in the next paper on nanowires. And this is what's so remarkable, is that the microbial world is able to perceive energy, and they've developed proteins that can actually take the electrical hit of something as potent as ionizing radiation and step it down to a level that they can then convert into chemical energy. And that's what's truly remarkable. We've all know about photosynthesis and photopigments, but here, what Arturo did with in the Chernobyl study is recognizing the use of energy by the microbe, albeit a fungus, is just phenomenal that they're able to do this. And it goes back to what's actually going on in the cells that we're going to talk about, where they're going to be dealing with oxidative burst, because that's nothing more than electrical hits as well.
Wow, this is very nice. I'm glad you brought that up. And I apologize for the digression, but I'm glad I did it. So, anyhow, let's go back to exocytosis. So here are yeast cells inside the macrophage, and they divide, and they grow happily. And after a while, and you can see this in the movies that go with these papers, out they pop. It's like, really, the cells goes, p'tui, out it goes. I don't want you anymore. And this happens with live cells. It doesn't happen as much, hardly any, with dead cells, and it doesn't happen with plastic beads. In other words, macrophages take up plastic beads, but they don't know how to spit them out. So it looks like, and other organisms don't do that. There's something specific about the organism, and of course nobody knows what that is yet, but I imagine that somebody will figure it out. What is it that causes the macrophage to be doing this? Okay, so anyhow, that's the story. It happens. The process can be delayed, as I said. The yeast can grow inside the macrophage for quite a while, but when it happens, it's almost instantaneous. I mean, it's within seconds or less, the exocytosis takes place. So anyhow, that's pretty much the story. The current paper deals with a way of studying this by using flow cytometry instead of using the microscope. It's an intricate technical paper. I commend it to the readers who care about how you study things, because this is a very good study on that. But the conclusions after that is that, in fact, acidification, which is a process that takes place after phagocytosis, has a role in this, and also that it takes place in vivo. They looked at macrophages from animals, not in culture, and the same thing happens. So this is a well-established phenomenon that had completely escaped me. I had no idea about this until I read about it.
So Elio, can you go back to the melanization thing? I mean, what was that connection?
Well, it's a little bit sideways. It is said elsewhere, not in these papers that I remember, that melanin is produced by Cryptococcus, and that is an antioxidant that helps the organism survive in the phagolysosome. But I made a big leap. I was just wondering, do the phagolysosomes are they black? Are they brown or black? No, I don't think you could. No, I don't believe so.
So the reason I ask is because in insects and in lots of invertebrates, the hemocytes go down, and there's an halide peroxidase that's related to the myeloperoxidase, and they when there's a bacterial infection, the macrophages encapsulate the bacteria, and then the halide peroxidase, there's a reaction that causes what they call melanization.
That's right.
It's so common, a sort of thing that happens. I was just wondering, it seems a bit like a highly conserved response. So I was just curious.
No, I'm sorry. I should have read more about it. I'm just not up on it. I don't know. I really can't tell you what the connection is with Cryptococcus, except that it's been mentioned. So we'll have to let it go at that. But it's something to look into, because this may be a very broad phenomenon, as you say, maybe highly conserved.
Yeah.
Margaret, what happens after melanization? Is that a step in the process?
Yeah. So actually, it depends upon the organism. And so, in the case, actually, it happens in the animal that I work on. So when they become septic, they take the bacteria are taken up by the gills as the blood circulates through the gills. They're encapsulated into little nodes, and then the hemocytes come around them, and they blacken the area around, and then the bacteria are killed, basically, and then the gills bud that off into the environment. In insects, it's a really big response, and it has to do some with the prophyloxidase cascade, something called the prophyloxidase cascade. So it all is, as Elio said, all about this oxidative stress environment that's created around the microbes. Yeah. And it leads to melanization as one of the byproducts of it.
In both those cases, the bacteria are the poor losers in the process. In this case, the fungi managed to escape.
Yeah. Well, so in the case of the insects and in the animal I work on, it's the host that's making the melanin.
Right.
And in this case, it sounds like this is making the antioxidant, right? Is that right?
That's right.
Yeah.
Yeah, that's the idea.
So it's protecting itself.
I wanted to say a couple things about this paper, if I could. I think that there were 2 points that I thought were particularly interesting. One is this paper really focuses on using this different technique, fluorescent activated cell sorting, to examine this process that people could see before, but in a very small scale using microscopy. So it really allowed them to use larger numbers to gain this information with new technology. But it also raises the issue that, in fact, you get some differences with the 2 different approaches. And I think that that's a really important concept, that each approach may have its own caveats that are problematic. And we may need yet a third approach to be able to sort through those things. So I thought that was really interesting. The other thing is that what this allowed them to do, this new technology, was to begin to examine the process. And when they examined the process, in fact, they found some pretty unusual results. And those results were that in some cases, it looked like you had a certain result when you neutralized the pH, and in other cases, you had the opposite result. And I think what that tells us is there's a tremendous amount we have left to learn about this process.
Can I ask how they neutralize the pH? So this is an intracellular vacuole, yeah?
They add a bunch of several compounds, chloroquine or ammonium chloride, and that does it. So
they did it the old-fashioned way, acid-base. They added base.
Right.
And in the other case, they added an ATPase inhibitor.
That's right, bacterium.
I see.
But those had the opposite effects, and what Stanley says is absolutely right. This says the process is complex, a little bit beyond the simple scheme that one would draw up for acidification and subsequent events in a fecal lysosome. And I'm not surprised that Cryptococcus may bring up new questions. It's just a different bug. It's a different sort of a parasite in many ways, and I think that's something that we should pay attention to. It's not the usual fungus infection. It doesn't cause the usual, but it's very different from bacterial infections. And so every detail about it may be kind of unique, and this exocytosis story points to that. I'm not saying that there couldn't be other organisms that don't do these things, but right now it's sort of, Cryptococcus is a bit of an outlier. Don't you think, Michael?
Yeah, I really do think it's a peculiar organism, and the one comment I was going to make to encourage Arturo's group to work on is to get his hands on, I'm sure he already has it, the Cryptococcus Gatdi from the Pacific Northwest that infects healthy people, and then run that organism through his macrophage assay with his elegant flow and ask the question, does it behave differently? Because I think that's the way we learn things, and that's the wonder of microbiology, in that with these cells you can oftentimes get strains with that subtle difference that causes a different phenotype in the host. And we know that Neoformans, for the most part, is avirulent in healthy individuals, while Gatdi can be quite virulent in healthy individuals. And the macrophage is not unique to healthy or immune-compromised individuals, they both have macrophages. So looking at that story and then isolating, because you can isolate macrophages from healthy and immune-compromised people and ask the question, what does the flow show? I think then we'll be able to begin to decipher some of these differences that Stanley so elegantly pointed out to us, and that Arturo and his group have really begun to tease through. And so I think this paper is going to be seminal for the methodology that they've offered to the field and the way the drugs are being added to, tinker with the pH and tinker with the biology of the macrophage to ask the questions, what is actually going on?
So one of the things that I'm curious about is, this is in the Pacific Northwest out in the woods. Where is it doing this naturally in nature? In other words, what was the situation in which there was evolutionary selection pressure on this behavior? So is it like the Legionella story where it was in an amoeba and actually what's happening out in the soil or wherever, hanging in the trees or wherever Cryptococcus normally is, are they typically in an amoeba and the amoeba spits them out?
It's a good thing you bring it up because Casa de Valle, in fact, studied the infection of amoebas, soil amoebas, by Cryptococcus, and the amoebas act just like a macrophage, or said more correctly, macrophages act like amoebas. So yes, you're quite right. This is probably the reservoir, or at least a habitat that Cryptococcus likes in the soil. It does live in, most likely in amoebas, and in that sense, it may have some similarities with Legionella.
Isn't it amazing?
If we're going to wrap this up, let me sort of summarize a few of the unique features of Cryptococcus. Is that okay?
That sounds great.
Okay. Well, okay. So it's a yeast. Other yeasts which are pathogenic, so that's nothing new. But it has a huge capsule, polysaccharide capsule. Now, others have two, but this one is particularly big. And it may have something to do with its pathogenesis, because if you look at a slide of tissue where it is rife with Cryptococcus, like in the brain or elsewhere, you find very little in the way of an inflammatory response. The tissue looks almost normal. Now, not always. There's sometimes when you find an inflammatory response, but sometimes you don't. So what's that all about? It causes disease without, it's a stealth organism. It does not set off the cytokine responses in the same level that other, I don't think at least, the way many bacteria do. Secondly, why is it so neurotropic? The meningoencephalitis you see in AIDS patients is the preferred site, although you find it infecting almost any other tissue at times, but the preferred site is the nervous system, the central nervous system. So that's all these things are fairly unique. And then now you add this ability of being able to, again, in a stealthy sort of a way, get into a macrophage and then get out of it and be happy.
Are there animal models of Cryptococcus?
Oh, yeah. You can put it in mice.
How far out? Is it mammals, vertebrates? In other words, I'm just thinking about there are some very interesting differences between the nervous system, say, of vertebrates and invertebrates. Obviously, vertebrate nervous systems are myelinated and invertebrates are not generally. I was just curious, this tropism may have something to do with the biochemistry of the nervous system.
It loves sphingolipids, I know that.
That's it then. No.
It loves sphingolipids.
See, one problem solved.
There you go.
Okay. Aren't you glad you asked? Okay. So it's a bug worth knowing about.
Thanks very much. I should have mentioned that this paper was just published in the journal MBio in the August 9th issue. So you can access that online. Check it out if you want to learn some more about Cryptococcus and the process of vomocytophagy.
Great.
The next paper we're going to talk about today, so a big theme in that paper was this impact of pH on the process. And the next paper we're going to move on and talk about electrons. This next paper is published in Nanotechnology in the 7th August 2011 issue. It's entitled Tunable Metallic-like Conductivity in Microbial Nanowire Networks. It comes from a large group, including the senior author, Derek Lovely. This is an impressive paper. It's from the University of Massachusetts, and one of the notable things here is that it is truly interdisciplinary. The authors come from the Department of Physics, the Department of Chemistry, the Department of Biology, and they collaborate with the Department of Biology at the College of Holy Cross. So a large group of people, and once we go through this paper, you'll see why they need to have this kind of interdisciplinary expertise. Michael, would you lead us through this paper?
I'd be happy to. This was a really delightful paper to go through because it allowed me to refresh my memory on that portion of physics that I took once upon a time that involved electricity, and also helped me to begin to reframe my thoughts about how electrons move within chemicals and specifically proteins. The paper starts off simply enough. They introduce the reader to this concept of pili. And any and all students of early microbiology will know that pili are that extracellular appendage that a large number of bacteria have. It's often composed of a single type of protein, and in this particular case it's called pilin. And it's not a very large protein. It's typically in the 130 type range. And what happens is these extracellular, if you will, structures are attached, and they facilitate everything from attachment to our epithelial cells. The pili associated with the gram-negative bacterium Neisseria gonorrhea allows the gonococci to hold on to our urethra quite well and result in an inflammatory response and all those other wonderful things. But what we learned as young microbiologists is that pili are beyond the resolution of the light microscope. Pili are typically 3 to 5 nanometers in width, and the resolution of a light microscope is about 200 nanometers. And so you can begin to see the challenge that is befalling these authors. And what they set out to do is to ask a very simple question. Were the pili associated with this one bacterium, Geobacter sulforeducens, could this extracellular appendage conduct electricity? And this is where it gets really neat. Derek Lovely's group has been working in this emerging area of microbial fuel cells and microbial energy, in which we're going to begin to try to harvest energy from our friends, the bacteria, and actually put it to useful work. And so Derek and his group for many years have been trying to decipher the mechanism of how these microbial fuel cells actually work. And it's really an exercise in electron transfer. And our understanding of how electrons move is going to be fundamental to our understanding of how he began to tease this process apart. So this group has access to a number of different types of Geobacter sulforeducens strains. Some make different types of pili, and he then begins to take us through this process of how he was able to demonstrate that pili do indeed conduct electricity. So we're going to have to, just like the last paper, we're going to have a digression about how electrons move. Now, consider that pilin, the foundation of pili, is a protein, and all proteins are composed of amino acids. So imagine that peptide bond where you have the carboxylic group interacting with the amide group. And again, refreshing your chemistry, remember that nitrogen is always a little partial plus, and oxygen is always electron withdrawing. So in my simple thought process, I think of electrons moving as flow, if you will, as a river moving downstream. So when you drop an electron into a river, it will flow with the current. And so imagining how electrons could move in a protein, you imagine how electron will bump in to it, and then through the aromatic resonance associated with the protein, you will then move the electrons through things. So we need a little digression about valence electrons, and for after all, that's how electrons flow in things like metals and semiconductors and in non-conductors. It all depends on how many valence electrons you have available. And so this valence electron concept is effectively where we begin to do the science associated with this particular paper. So,
Michael, I got to interrupt you. I think that TWiM should raise your pay right here. You've done such a good job of explaining that. But I have a question. How much of what you just said is just, let's say, paper electrochemistry, and how much is something that you can prove in general terms?
Well, this is what this paper is all about. He is actually going to prove that the electrons are moving through this protein. Foundationally, at an atomic level, you need a quantum mechanic to begin to explain this. And this is sort of jumping ahead to one of my next digressions, and that's he introduced me to an old term that I had forgotten about, but then had to relearn when I began to take this paper apart, and that's something called a phonon. And a phonon, it's this, if you will, excitation. When an electron bumps into something, it effectively allows the entity, whether it be a protein or an atom, take the electrical hit. And going to my favorite source for physics these days, Wikipedia, a phonon is operationally described as a collective excitation in a periodic elastic arrangement of atoms or molecules in condensed matter such as solids and some liquids. Well, I always imagine proteins as being elastic in that they're able to form and fold into different shapes based on the pH and their local microenvironment. And in the elegant study that he describes with his microbial fuel cells, he uses a split gold anode. And again, think about what's going on here. So I'm going to start very simple. He's growing these Geobacter on 10 millimolar acetate, a 2-carbon compound. It's effectively an acid. So we all remember our Krebs cycle. It's, what comes out of the Krebs cycle. After you make pyruvate, you blow off the CO2 and you have the acetate, and then the acetate feeds into the Krebs cycle, and it gets burned ultimately to CO2 and water. Well, in aerobes, they dump the waste electrons into molecular oxygen, and the net consequence is they end up making water. But in the majority of microbes on the planet, molecular oxygen is typically not available to dump those waste electrons. And so you could have an infinite carbon source, but unless you have a place to put your waste, you will not grow. And so that's what these fuel cells are all about. You grow the Geobacter on this acetate, and the electrons have to go someplace. And where they go is the anode. And the anode
was an electrode. An electrode,
it is an electrode,
like in a battery.
Right.
The anode, the mnemonic I learned when I took my electrochemistry once upon a time, is the mnemonic for an anode was acid, which is the anode and the current goes into the device. So the anode goes in, the electrons go into the anode, and then, as he describes in the cartoon associated with Figure 1, you can actually see his diagram, and the electrons go up a wire over to the cathode. But like everything in biology, it has to be done in a pair. So if an electron moves, a proton similarly has to move. And that proton is excluded from moving to the cathode by this impermeable proton exchange membrane. And so what will happen is the electrode, the split anode, effectively transfers the electron up through a wire, so you're having current move. It goes to the cathode, and then the proton comes across the proton exchange membrane, and it then revisits the electron, and you end up making water at the cathode. And oftentimes the cathode is in an aerobic zone where you have the availability of oxygen, so the electron comes, you effectively make the half-cell potential, and the net consequence is the circuit is completed. So Derek and his group...
This is new, actually.
What's new is their hypothesis of the paper was that the network of pili nanofilaments might confer the conductivity to Geobacter sulfur-reducens and their respective biofilms. And so what they did in this paper is they manipulated...
Michael, before you go on, I think that their control here is pretty important.
Okay.
And this might be a good time to say something about that control because it eliminates the chemical possibility here, right?
Okay.
So I'm talking about the control where they have the effluent going out with the excess substrate into the separate container that does not have the split anode.
Okay. And he's not seen any conversion of current.
Right.
And he does that with a very elegant series of, he shows you what is actually going on with his confocal microscopy, showing how the biofilm grows over the split anode, and in the control situation where there is no circuit completion to the cathode, it doesn't happen. So that's actually a very good point, Stan. Thank you. The other thing that another control that's associated with this is when they grow the microbe on fumarate, which is an accessory electron acceptor. The microbe is able to dump its waste electrons to the fumarate, and it no longer has the need for the cathode, and hence the current levels drop, and you do not see the same level of conductivity that you would anticipate if the circuit was being completed. So that's actually another very elegant control when you provide the microbes with an alternative electron acceptor. They don't need to use the microbial fuel cell as their electron transferring device, and they actually downregulate the amount of pilin that is being produced. The other thing is they also have controls with Pseudomonas aeruginosa in E. coli, and these organisms develop biofilm, and they grew across the non-conducting gap. They, too, their conductivity was not significantly greater than the control without a biofilm.
It's interesting that this can span the microbial world pretty nicely. I seem to remember that Yuri Gorby found that Cynecocystis, which is a cyanobacterium, a photosynthetic cyanobacterium, can make electricity by using light. In other words, the power of photosynthesis is changed into electricity. You remember that?
Yes. It's all about moving electrons. And I think if we just keep that concept central in our thinking as we navigate through this paper, we'll see the elegance that this group has been able to actually demonstrate that in this particular microbe, it's the pilus that is transferring the electrons. And one of the experiments, and I apologize, I haven't had time to get into the literature to know whether or not his group has done this experiment, is to take the pilin gene out of Geobacter and put it into E. coli or Pseudomonas, and then ask the question, when you put that gene, so you're just making pili in E. coli and Pseudomonas, whether or not electrons would be transferred with this organism that's able to form a biofilm with all of its other genes, and whether or not it would then actually serve to conduct electricity. I think that would, doing the experiment. As he takes us through this paper, he does this both in situ with the biofilm, and then he goes to that old experiment that I did as a graduate student where we took E. coli that actually produced pili and we put them into a Wearing blender to shear off the pilus in order to be able to measure the protein concentration and do all those other things. And he used purified pilin to actually demonstrate a lot of the electrochemistry associated with these, in addition to doing it with the biofilm, as well as doing it with a series of mutants. The other thing that's really neat is in Figure 3, he actually inactivated some of the cytochromes that were in the membrane, because some of his critics to actually, this was a controversial subject till probably this paper, in which people were arguing that the cytochromes were actually moving the electrons to the anode rather than the pilus. Lovely's group has been arguing for the pili being responsible for moving it, and I think this elegant paper has now demonstrated that it looks like the pili are indeed moving the electrons from the metabolic event of oxidizing the acetate, the anode, and ultimately generating current.
Isn't one of the interesting things in the paper that by changing the gene expression, they could change the conductivity involved?
Right, and that was with fumarate. He did that with fumarate, as well as with KN400, which is a mutant that he was contrasting against DL1, which was another strain, and a fumarate grown DL1 that has lower conductivity. So all of that is in Figure 3A. It's a pretty neat story that he takes us through there.
But there's a real important point here, and that is there's this correlation of pili with conductance at this stage of the paper so far, but it doesn't prove that the conductance is via pili. So the pili could influence the bacteria coming together. It could influence how they come together. It could influence other factors in the outer membrane. It could influence a lot of things, right? This, so far in the paper, not yet proof.
That's right. And that's where he then takes us to Figure 4, where the title of Figure 4 is the observation of metallic-like nature of conductivity. And here in his central hypothesis is the network of pili might convert conductivity to the biofilm established by Geobacter. And so he shears off the pili, and he has a purified prep of the pili, and he dries them down, and he wets them, and he does all the appropriate experiments. In addition, he grows up the biofilm, and then just like you peel off a fruit roll-up, he peels off the biofilm that he's grown, and he deposits it down onto the set of gold electrodes that he's then able to measure. He calls it a four. I want to get it right. He calls it a four something or other, a 4 gate. No, not gate. As I said, this was a tour de force to get through. But he measures it at 4 spots, and he's able to measure the conductivity of the purified protein as well as the biofilm. And what he was able to show is that the purified pili filaments had approximately 3 orders of magnitude higher conductivity, and that this conductivity was dependent upon temperature. And as he said, is that the temperature was important for the conductivity. Conductivity began to decrease exponentially upon heating at temperatures where the thermally excited backscattering phonons began to scatter the charge carriers. So he was able to go and demonstrate that it was indeed behaving as a quasi-metal, if you will, or as he referred to it, as a quasi-one-dimensional organic metal, rather than the linear observation he anticipated.
I think that's a really important experiment that is a more direct test of the hypothesis.
But I think he had to lay all that groundwork before he got to figure 4. He had to demonstrate it, that you could change the expression by adding fumarate, so to demonstrate the biological importance of the pilus being the principal conductor. The organism would make this when it would need to be able to dump its waste electrons.
But not to be the devil's advocate, but all this proves that pillin is essential, but it doesn't prove that it's the only component, does it?
Correct. He was just going out to prove it was conductive.
Okay.
He really wanted to demonstrate that it was a conductive. He's been taken to task because he has maintained for quite some time that the pili were the principal way the electrons were getting to that anode.
I think that the point is that for many people this is a big surprise because pili are composed of amino acids, and amino acids, as a rule, are not very good conductors of electricity. And our proteins in general aren't great conductors. And so this particular case, I think, was a surprise to a lot of people. And so many people wanted to develop alternative models. And in fact, I think it's still possible that in other related organisms, there are alternative ways of achieving a similar result.
Yes, and I actually think it comes down to he's going to have to go and elicit some of the skills of molecular biologists to begin to change the composition of the pilus in Geobacter and get rid of some of the aromatic moieties in that 100 mer base building block for pillin. He's going to have to begin to slice and dice to change that to, again, see the subtleties and how much current can actually flow through that nanowire.
Michael, did you say that they have an unusually high proportion of aromatics, pillin proteins?
I looked at that. I looked at the sequence and I began counting them, but then I ran out of time. It didn't seem any higher. There's a large number of charged amino acids in the pilus. It's 137 amino acids in length. It's secreted via the type 2 secretion system. So being an old type 2 secretion biologist, the first experiment I would do is I would add azide to the system to knock out type 2 secretion, because that will poison the Sec-A molecule, and you won't get any type 2 secretion, so you won't have any pilus. But we sort of know that the mutants without pilus don't conduct, so I killed that experiment in my mind right there.
Besides, azide does a few other things.
Yeah. In fact, with these mutants, it's a little tricky, and this is one of the complexities of working with bacteria that aren't model systems. I don't believe that these mutants are isogenic strains. So what that means is that we know they have this difference in the amount of pili, but we don't know what other differences they might have that could be important. And so it gets very complex. And that's really one of the keys of an elegant genetic system, is that you can narrow things down to one particular component.
I look to see whether these strains have been sequenced. The entire genome is known. It's up on GenBank, because I did ask that question to see if I could do an in silico experiment to ask the differences. But as everyone knows, you can't do that in 20 minutes. And I think it's a very important point that Stan brought out, that they are not indeed isogenic strains.
So are they thinking that this is a common characteristic of the pili that are in biofilms across the board?
No, because Pseudomonas, as we all know, forms a tremendous biofilm,
right.
And it's non-conductive. But again, Pseudomonas is an aerobe, so does it need a conductive pilus?
Okay. Is it an aerobe or is it a facultative anaerobe? Pseudomonas.
Geobacter sulforeducens. It's growing in the dirt.
Okay.
My question, Michael, is when do I need a culture of Geobacter to turn on the light in my living room? How far along are we from that?
That's the great debate, and the utility of microbial-based energy generation. I think, it's all about how much current you get and how many watts and how many amps. And I guess a lot of folks are trying to figure out how to scale these systems up. And at the end of the day, we have a global energy budget that requires plants that generate gigawatts of energy, as opposed to the microwatts that the microbe is generating. But we can make, giga quantities of microbes, so if we're able to figure out how to move the current and not lose it to friction, again, that may be the thing to do. And it all goes back to that phonon that he brings up. And the phonon is really, the electrons or the charge transfer bumping into one another in that quantum mechanical space. And so if you don't lose your energy to friction of the things bumping into one another, you may be able to look at these pili as, if you will, superconductors, but it doesn't appear they have superconducting properties where you could get a lot of energy without losing energy. So the answer is, I don't know when you're going to be able to.
Not tomorrow.
I think the answer is don't hold your breath.
Right.
Yeah. Well, thank you very much, Michael. That was a pretty complicated paper, but it's exciting to think about this kind of different function that microbes do that has a lot of potential for the future. With that, I'd like to remind readers to email us, send your questions, comments to TWiM at twiv.tv, or leave a comment at microworld.org/TWiM. And we'll read a few emails from previous sessions. First, Michael writes in response to TWiM number 9, Specimens for Undergraduate Labs. I am a student that is just starting the 2-year professional phase of my medical laboratory science degree. And from all I know, when possible, many of the samples that program uses are actually from 2 major local hospitals. However, this is not just any undergraduate course. Some of the lectures can be taken by students in molecular and microbiology major, but the labs are restricted only to the medical laboratory science majors. By the way, I love TWiM. I started listening to TWiV in early 2010. I enjoy it even more that I can now listen to you at 2x speed, and therefore I get 2x the info.
Oh boy, we better talk fast.
Or talk slow.
Or slow.
One suggestion for a future topic. It would be great for you all to discuss how the advances in technology will be able to change the time and types of medical testing. Techniques such as PCR, flow cytometry, and maybe even microarrays, to name a few. Thank you again, Michael.
I'd like to comment to Michael on this excellent comment and take up his suggestion. One of the things I'd like to see us discuss on a future TWiM is the use of MALDI mass spectroscopy for the rapid identification of clinical specimens. At the recent general meeting for the American Society for Microbiology, a large number of our clinical vendors were displaying these systems that employ MALDI mass spectroscopy, and they've been able to get the identification of a microbe down to 10 cents a shot, and it's real time, and you can identify the bacterium pretty quickly. So this was a technology that was also came out of ONR, and for the rapid identification of infectious processes from people at sea. And so I think that whole area of rapid identification of microbes is really pretty exciting.
I think then there's the next level, the generation that we haven't yet achieved, which is the potential advances in solid-state technology and laser technology that may make even that wonderful approach outdated before long. So it's pretty exciting time in the development of technology. I think with flow cytometry, the paper today on Cryptococcus really emphasized how this approach can increase your throughput to allow you to see things that you wouldn't be able to see using previous technology. Next letter, John writes, Dear Twimmers, I love the pick of the week for the life at low Reynolds numbers. I originally read this a few years back and was recommended alongside of a couple of other papers that I thought would make good listener picks of the week. Both papers are about scaling in biology and why, for example, ants can't grow to be as big as a dog. First, on Being the Right Size by Haldane. This is a classic paper everyone should read. Second, Biology of B-Movie Monsters by Michael LaBarbera. Thanks again for your wonderful podcast. Both of those papers, I should note, are available online.
They're both terrific. They're both an incredible read. It gives you an idea of what the physical and mechanical limits are to life and to developed life especially. So we all have an interest in, we see our own bodies in reading these two papers. Wonderful stuff.
And the Haldane paper was originally published in 1928.
Right.
And so I'll have our friends at ASM post those two papers in the show notes for us.
That's great. Now, I haven't read about the biology of B-movie monsters, but with a title like that, I'm going to go read it. So,
thank you, John.
Another paper, Rufus writes, the aside regarding what it would be like to swim in corn syrup is actually a rather old question. Drag certainly goes with viscosity, but viscous drag may not be the speed-limiting factor for human swimmers in water, and propulsion efficiency may go up with viscosity over some range, say, ethanol to water to pudding. A chemical engineer decided to test it for humans in a liquid with double the viscosity of water, using one of those old 4-lane swimming pools. With double the viscosity, swimmers could maintain the same times. Note that in another secondary reference to the paper, though they could maintain the same speed, athletes reported more fatigue from the viscous swim. The reference to this is a paper entitled, Will Humans Swim Faster or Slower in Syrup by the American Institute of Chemical Engineers Journal, Volume 50, in October 2004, page 2646. Sounds like an interesting paper. Interesting experiment, actually.
I don't think I'd want to be one of those swimmers, though.
Probably pretty sticky when you got out of the pool.
Uh-huh.
I suppose our intuitions are even more suspect when looking at navigation and propulsion at the micro and nano scales. Rufus Knapp, SW Engineer, Portland, Oregon. Thanks a lot for that input, Rufus. And then the last email of the day, Dave writes, I found the discussion of two papers in TWiM number 11 fascinating because you addressed the incredible complexity of biological systems and the immense difficulty of testing hypotheses of a simple cause and effect nature. As a person with 50 years of experience in mathematical modeling and statistics, I have great appreciation of just how difficult it must be to design valid experiments and models to test almost any biological hypothesis. However, your discussion of entropy in response to a letter from one of your listeners seemed to wander off in strange directions. Think the problem started with the letter writer misstating the key component of the second, not the third as stated in the letter, law of thermodynamics. Entropy, or disorder, increases in a closed system, not in any system, as the letter writer stated. The biological examples he named that appear to counter this law were open systems. These biological entities are obviously exchanging energy and matter with the greater universe. This is what drives self-assembly and other apparent violations of the second law of thermodynamics. I'm sure that any closed biological system that one could create would obviously run downhill from an entropy standpoint. Thanks to you and your other panel members for great podcasts. Listening to TWiM, TWiV, and TWIP is the high point of my week. Dave.
How nice. I think that clarifies a lot, and I think it's a complicated subject that we may or may not want to go into in the future, but certainly the misconception that biology negates the second law of thermodynamics has been put to rest many times, and I'm glad to see it put to rest once again. I think that Dave explains it very well.
Absolutely. That was a very nice letter. Thanks a lot, Dave. So we'll wrap up. I'd like to point out that there are many ways to listen to TWiM at iTunes, Zune Marketplace, or consider subscribing. It's free and allows you to automatically receive each new episode as it's posted every other week. You can also listen with Microworld app streams episodes to your iPhone or iPad. Check it out at microworld.org. It's now available for Android devices at http://bit.ly/mwandroid. You can always download episodes at microworld.org/TWiM, where we also have show notes. If you like TWiM, please tell others about it. Links and reviews in iTunes are particularly helpful for keeping us up there on the top of the page. Questions? What do you want to hear? Any other comments that you have? Please make your comments on TWiM at twiv.tv or go to microworld.org/TWiM and leave your comments. If you have a story or you would like to submit some idea for discussion on TWiM, add it to microworld.org and tag it with TWiM. Thanks very much to everybody. Thank you, Michael Schmidt at the Medical University of South Carolina.
Thanks, Stan.
Elio Schaechter is at Small Things Considered.
Thank you so much.
Margaret McFall-Ngai at the University of Wisconsin.
Thanks, Stan.
And I'm Stanley Maloy. I'm at SDSU. I'd like to thank ASM for their support of TWiM, Communications Director Barbara Hyde and Chris Condylan, and Ray Ortega for organizational and technical help. And thanks to you very much for listening. We'll see you next time on This Week in Microbiology.